Okay, recording. Yes. All right. Let's finish up the digestion system, digestion 3. Left off here, chemical digestion and absorption. We'll look at the nutrients that that we eat and how they are broken down, how they are absorbed, and this will lead us right into the nutrition chapter. Okay, carbohydrates, simple carbohydrates. I don't think they should be called carbohydrates, but on food labels they're called simple carbohydrates. Mono and diglycerides. Sugars like glucose, sucrose, fructose, maltose. Okay, complex carbohydrates are at least 4 sugars in a row. And there's 3 different ones: starch, cellulose, and glycogen. Starch and cellulose are plant products. Glycogen is also called animal starch. Okay. Digestion. So when we eat starch, which is a polysaccharide, we digest it to oligosaccharides, which are just a few glucose monomers long, and then we digest it to disaccharides and then to monosaccharides, and that's how they have to be absorbed as monosaccharides. And by amylases, either salivary amylase or pancreatic amylase. Any monosaccharides absorbed other than glucose are converted by the liver. Now remember, these sugars go directly to the liver along the hepatic portal vein, and the liver converts them to glucose. So if you have fructose, galactose, it's converted to glucose, because glucose is blood sugar, and it's the most important sugar, and it is the sugar that complex carbohydrates are composed of. Okay. So there's the enzymes that do it. Salivary and pancreatic amylases, these brush border enzymes with dextrinase and glucoamylase do it. And as you mentioned, milk and table sugar are digested by lactase and sucrase. And this thing about lactose intolerance, some people are lactose intolerant, does that mean that they don't have lactase? Well, everybody's got lactase, the gene for lactase. It's just turned off. Well, early in our evolutionary history, it was turned off after weaning, after breastfeeding, and then we never touched the stuff ever again. And that's how most mammals are. We are really strange mammals in the sense that we drink milk after we're weaned. No other mammal does that. And even stranger, that we drink milk from some other mammal. No other mammal does that either. So anyway, in some people that gene is turned off and therefore lactose intolerant. Fatty glucose. What happens to glucose? It goes into your body. It's put in the blood and used immediately to feed cells, or stored as glycogen. And this is our storage carbohydrate, glycogen, stored in liver and in muscle. And if you eat more sugar than you need to fill up your glycogen gas tank, which is finite, you make fat out of it. You can make fat out of sugar. And there's just a figure right out of the book, talks about these pancreatic amylase, brush border enzymes, lactase and sucrase, and then pancreatic amylase breaking starch into oligosaccharides, glucose, maltase, and then those other brush border enzymes converting them to monosaccharides. And remember, that's how we absorb it. Proteins, which starts in the stomach with pepsin. That's converted from pepsinogen by the hydrochloric acid in the stomach. Proteases is an enzyme that breaks down protein, digests the polypeptides and oligopeptides. So polypeptide really is a protein. It's like a polysaccharide is a carbohydrate. And it's similar to the carbohydrate digestion, to oligopeptides, which is the few amino acids long. And aminopeptidase digests the dipeptides. Where dipeptides is 2 amino acids. Remember, that's the monomer for proteins, amino acids. And we have to digest protein into individual amino acids in order to absorb, to get the benefit from it. That's how we absorb the breakdown products of protein amino acids. Just like starch, carbohydrates have to be broken down to glucose, proteins have to be broken down to amino acids. And there's brush border enzymes there also. Some whole proteins taken in by endocytosis. This is really rare. When you're an infant getting back to breastfeeding, you get some antibodies from mom's breast milk, and those are exocytosed. There's a couple other ones I'm going to get into that's pretty rare. Okay. Fate of amino acids. Well, your body can use them to make proteins. Or you can convert some amino acids into a glucose by a process called gluconeogenesis. This only really happens if you either run out of carbohydrates or you don't need any carbohydrates. You can also convert amino acids to triglyceride, fat, and store in adipocytes. And even though proteins are caloric, that is, you can get calories from it, you can break it down to ATP. We generally don't do that unless we eat way more protein than we need to make our to make new cells, to make proteins, to make enzymes, to make hormones. If that's the case, then we usually convert it to either carbohydrate or fat. There is no storage form for protein. So we'll talk about calorigenic things you can make, get calories from. Three macromolecules that are calorigenic are carbohydrates, fats, and proteins. And we have storage forms of carbohydrate and of fat to keep us going in between meals. There is no storage form for protein. If you don't eat for a very long time, you will start breaking down your own muscles to keep your brain alive. But that only happens in starvation. And there's the, again, a figure right out of the book, talks about all these protease enzymes. Remember, the protease enzymes, they're always made in an inactive form, as most of them have that ogen in the end, and they have to be acted upon by other enzymes, or in the stomach by HCl, to break off that ogen and to make them active. So trypsinogen, chymotrypsinogen, procarboxypeptidase all come from the pancreas, and they're converted to carboxypeptidase, chymotrypsin, and trypsin by these brush border enzymes like enteropeptidase. And then, like I said, they break it up into dipeptides and then dipeptidases. Break the dipeptides into individual amino acids, and then we absorb. Oh, wait a minute. The names kind of tell you where these proteases work. A carboxypeptidase acts from the carboxyl end of the protein. Aminopeptidase works in from the amino side of the protein. Endopeptidases work in the middle of the protein. Okay, lipids. So I have lingual lipase, gastric lipase in the stomach, and pancreatic lipase. Now, in order to effectively, or what's the word I'm looking for? To efficiently absorb the breakdown products of fat, you have to emulsify them. That is, break these big fat globules into very small globules, increase the surface area for the lipases to work. And bile, that's one of the major functions bile and bile salts do. They emulsify. And the triglycerides, which is our dietary fat, triglycerides, they are broken down by lipases to monoglyceride and free fatty acids. And these are absorbed in these little phospholipid balls that's also put into the GI tract along with bile from the gallbladder are the little balls of phospholipids, and they absorb the monoglycerides and free fatty acids. Then these micelles are brought into the enterocytes right across the cell membrane, because they're like cell membranes themselves. And the triglycerides are reconstituted inside the enterocytes. Now, enterocyte is the cell that lines the small intestine. And packaged into these protein packages called chylomicrons. And these chylomicrons are exocytosed on the basal part of the endocyte, put in the interstitial fluid, and they are too big to fit into capillaries, so they're put into the lymphatic capillary. The lacteal, that's there's a lacteal in every villus, and the chylomicrons are put into the lacteal. They percolate down to lymph nodes and everything in the thoracic duct, up to the subclavial veins. Remember, they're sent to the subclavial veins, and they end up going directly to adipocytes. Remember, dietary lipid is the only nutrient that doesn't go directly to your liver via the hepatic portal vein. They go directly to adipocytes. They don't go to your liver. Transported in these chylomicrons. And so a chylomicron is a type of lipoprotein. Because we're going to see a couple other lipoproteins later on when we get into nutrition. And here's, again, right out of the book, this figure here talks about the bile salts released from the liver and emulsify lipid droplets to form micelles. Pancreatic lipase functions within the micelles to break triglycerides into monoglycerides and free fatty acids. These micelles are brought into enterocytes. The triglycerides are reconstituted, they're put into chylomicrons, exocytosed into interstitial fluid, into the lymphatic capillary. Nucleic acids. We need nucleic acids too, but they're not energy yielding, or not caloric. We don't use them for energy. We just use the nucleotides, and of course, you have to break the nucleic acids into nucleotides and bring those in. And then we can use those nucleotides to make our own nucleic acids. There's deoxyribonuclease and ribonuclease. Break nucleic acids down to nucleotides, and then a nucleotide breakdown by brush border enzymes called phosphatase and nucleosidase. So I guess we absorb them as the phosphate, the nitrogenous bases in the ribose or deoxyribose. Okay, vitamins, absorbed unchanged. So we just absorb them across into the enterocytes. The water-soluble ones absorbed by diffusion, and there has to be carriers, membrane carriers for these because they can't cross the cell membrane. They're water-soluble. The only exception is for this is vitamin B12. Remember, it has to be attached to intrinsic factor in order to be absorbed. So it must be endocytosed, because that would be way too big to just diffuse in. And so remember, the intrinsic factor is made in the stomach by cells in the stomach, and it's absorbed in the terminal ileum, the last part of the ileum. Okay, minerals. A lot of minerals are co-transported with sugar and amino acids. Sodium is co-transported with sugar and amino acids. Remember, this is called symport. This is secondary active transport, where the sodium concentration gradient is used to transport things into the cell, particularly sugar and amino acids. Minerals, potassium by simple diffusion. And most of these ions, like sodium, chloride, potassium, you will absorb into your body as much as you put into your mouth. So you can actually OD on this. You can actually take too much sodium or potassium to be dangerous. There's one guy, this guy, I read about this kid who on a dare drank a whole bottle of soy sauce, the equivalent of 175 grams of sodium, of salt, all at once. Well, they had to rush him to the emergency room, pump his stomach, just barely saved his life. He almost got Darwin, because as much sodium as you put in, you're going to absorb all of it. Right? Same thing with potassium. But iron and calcium are different. They're controlled. You only absorb enough iron to fill your body's needs, so you can't OD on it. There's a hormone called hepcidin that's put out by the liver that inhibits the movement of iron from the enterocytes into the blood. You see, if you eat a huge amount of iron, you will absorb it into the enterocytes, but it won't get into the blood because hepcidin is there. In order for it to get into the blood, the hepcidin has to be removed. Then the iron can enter your blood. So if you eat way too much iron, though the iron will stay in the enterocyte, and eventually that enterocyte will be sloughed off and end up part of feces, and you'll avoid the excess iron that you ate. The iron is transported when the hepcidin is not present. The iron is transported in the blood by a hormone called transfer. Calcium, same thing. You need PTH, parathyroid hormone, in order to absorb calcium. And so you can't OD on calcium either, because it's controlled by it's a regulated absorption. And it's absorbed by diffusion, and it can be actively transported. Yeah, there's the calcium parathyroid hormone, and vitamin D also increases calcium absorption. Okay, water, mostly absorbed by osmosis in small intestine. And remember, it's one of the major roles of the larger intestine is to absorb water. Just enough water, and the bottom part, diarrhea and constipation. You don't absorb enough water, it's diarrhea. Absorb too much water, it's constipation. So you want to have just the right amount of water. And remember, so that's one of the losses of water, about 200 milliliters a day in feces. And that's it, folks. For the digestive system. So we're all done, and then moving on to nutrition. Okay. I'll see you later.